All right, alrighty, so let's talk about how to translate this DNA sequence into an amino acid chain. So the very first step that we have to do is translate this DNA strand into mRNA, and the M stands for messenger in this case. All right, so to do this, we need to know our base pairing rules to get from DNA to RNA, and here are the base pairing rules. A will always turn into U, T will turn into A, and G and C will always turn into each other. So I'll do this one in blue. So our first letter we have is T, and according to our base pairing rules, T turns into A, and then we have A turns into U, and then C turns into G. Then we have T, which turns into A, G, and then U, G, A. All right, so that is how we translate the DNA strand into the mRNA strand. Now, the next step is to turn our mRNA strand into an amino acid chain. Oops. All right, so the way that we turn mRNA into an amino acid chain is by using our codon wheel over here on the right. So I'm going to highlight and show you how we do this. So our very first codon, we have A. Oops. Where's the highlighter? There's the highlighter. Okay, our very first letter, we have A, right? So I'm going to go over here to my codon wheel in the very center, and I'm going to find A, which is right here. Let me use a different color. I'm going to use A, which is right here, and then I'm going to go to my next letter, which is U. I'm going to find U, the next one down, which is right here, and then I'm going to find G, which is right here. Now, the amino acid that we see right there is MET, so I'm going to write MET below AUG. And then for the next one, I have an A, so I'm going to go to A again, and then I'm going to find G, which is up here, and then I'm going to find G again, which is right there, and we see that amino acid is ARG. And then for my last one, we start with U this time, so I'm going to go over here to U, and then G, and then A, which is stop. And I'm going to write stop right here. Now, what you'll notice is in the beginning we have MET, and MET has a little start next to it. Almost every single amino acid chain will start with MET, and it will stop with one of the 3 stop codons, because that tells the mRNA when to start coding and when to stop coding, so the protein is the correct length. So, again, the very first thing you have to do is get your DNA strand, and you have to translate that into mRNA, which you do with these base pairing rules up here, and then you use the codon wheel to turn the mRNA sequence into these amino acids. Hope that makes sense.